A healthy-looking field can harbor an invisible crisis. Microscopic invaders often multiply quietly within plant tissues, destroying an entire harvest before the damage is even visible on the surface. When rot or wilt appears, the immediate instinct is to blanket the field with chemical treatments. However, blind applications rarely stop the spread and waste valuable resources. Saving the crop requires tracing the symptoms back to a specific biological culprit. This diagram maps out the 4 primary categories of plant pathogens: fungi, bacteria, viruses, and nematodes. Each group possesses a unique biological trait and requires a different testing method to reach an accurate diagnosis. To determine which one is attacking the field, we build a diagnostic profile. By systematically moving from broad field observations down to precise microscopic analysis, we can isolate the threat and stop its progression. The process begins in the dirt. Before taking physical samples back to the laboratory, we perform a visual inspection of the entire living plant in its natural environment. Here we have a direct comparison. On the left is a smooth, healthy green leaf. On the right, we see distinct leaf mottling and yellow, mosaic-like chlorotic patches. These are clear visual indicators of a viral or viroid infection. It is crucial to look at the distribution of these symptoms. Fungal infections typically create localized necrotic spots right where the spores landed. Viruses, by contrast, infect the vascular system, causing systemic deformation like severe leaf rolling and crinkling across the whole plant. Diagnostic accuracy at this stage is vital. Complex viral mottling is frequently mistaken for simple nutrient deficiencies in the soil, such as a lack of nitrogen or magnesium. Because viruses hijack the host's internal machinery to replicate, they consistently stunt the overall growth of the plant. Verifying this systemic stunting is a mandatory first step. If the symptoms remain localized, we rule out viruses and move on to physical soil extractions. If we find root damage, like distinct galls or stunted lateral roots, but the plant lacks systemic leaf mottling, we check the surrounding soil for plant parasitic nematodes. Since these parasitic worms are microscopic, we isolate them using a wet sieving process. We mix the suspect soil with water and pour it continuously through a vertical stack of metallic laboratory sieves. This cross-section shows the filtration logic. Top meshes, 500, 200, and 100 micrometers, trap coarse debris. Nematodes slip through to the final 63 micrometer layer. To isolate living targets, we transfer this catch to a Behrman funnel. Active nematodes wriggle through a filter, collecting at the bottom. Precision is required here. If you pour the slow water mixture too aggressively, the sieves will overflow, washing away the microscopic worms and ruining the sample. Physically isolating these worms from the dirt confirms that root destruction is a parasitic attack rather than a result of poor drainage or soil chemistry. In the laboratory, we check for bacteria. If the plant shows distinct cankers or rapid wilting, we take a tissue swab from the infected area for a chemical gram stain. The chemical sequence follows 3 steps. First, we apply the tissue swab to a glass slide. Second, we flood the slide with purple crystal violet dye. Third, we set the dye in place by adding drops of Gram's iodine. Next, we apply a brief acetone alcohol wash. This acts as a stress test for the thickness of the bacterial cell walls. On the left side are Gram-positive bacteria. Their thick cell walls retain the initial dye, keeping them a distinct purple. On the right, Gram-negative bacteria have thinner walls. They lose the purple dye during the wash, taking on a pink or red appearance. Timing is critical during the wash. Leaving the acetone alcohol on for a few seconds too long will bleach the slide entirely, leading to a false negative. This chemical reaction divides the bacterial kingdom into 2 color-coded categories, rapidly narrowing down the list of potential culprits. We then examined fungi, the most common cause of plant disease. Accurate identification requires 2 parallel tests: microscopic measurement and active agar plate culturing. First, we examine a sample under a light microscope to document its physical anatomy. We record the spore shape, measure its size, and document the branching patterns of the structures that hold them. In parallel, we run a culture test. We swab the sample onto a selective nutrient medium, like V8 juice agar, and incubate it to observe its growth behavior. This time-lapse shows the diagnostic evidence. A fuzzy fungal colony radiates outward from the center of the plate, demonstrating how the pathogen spreads. The microscope identifies the physical anatomy of the spores, while the culture plate reveals colonization patterns. Combining these data points allows us to pinpoint the exact fungal genus. We have now built a complete diagnostic profile. By combining field observations with nematode sieving, bacterial stains, and fungal cultures, we reveal the specific identity of the pathogen. With a confirmed identity, the uncertainty ends. We can now implement a targeted, integrated pest management strategy. This data dictates the response. It allows us to select genetically resistant crop varieties, introduce specific biological predators, or apply targeted chemical controls that only affect the identified pest. Precise identification provides the technical foundation for agricultural security. By isolating the specific cause of decay, growers can preserve their current yields and prevent the long-term degradation of their